This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we're diving into the captivating world of bats, specifically 3 remarkable species that will change how you think about these nocturnal mammals. We'll meet the spectral bat, a powerful predator of the night, fishing bats that snatch prey right from the water, and the notorious vampire bat, whose real story is far more fascinating than folklore suggests. Each of these extraordinary creatures has evolved unique abilities that push the boundaries of what we thought bats could do. So settle in and prepare to be amazed by nature's ingenuity. Chapter 1. Acoustic Mastery of the Night Sky. When the sun sets and visual hunters retreat to their roosts, a transformation occurs in the night sky. The aerial domain, teeming with insects and small vertebrates, becomes accessible to a group of mammals that have evolved one of nature's most sophisticated sensory systems. Bats, comprising roughly 1/5 of all mammal species with over 1,400 species worldwide, have developed echolocation to such a refined degree that they can detect, track, and capture prey in complete darkness with precision that rivals or exceeds what daytime predators achieve with vision. The physics of echolocation operate on principles both elegant and complex. A bat produces ultrasonic calls ranging from as low as 5 kHz to as high as 200 kHz depending on species, though most operate between 20 and 120 kHz, well above the range of human hearing. The use of ultrasound rather than lower frequencies reflects fundamental physics. The sonar cross-section of a target, which determines how much sound energy reflects back to the bat, scales with the inverse fourth power of wavelength. Small insects with body dimensions measured in millimeters create much stronger echoes when ensonified with high-frequency ultrasound than they would with lower-frequency sound. This relationship between frequency and target size explains why bats converged on ultrasonic echolocation for detecting prey that would be nearly invisible to lower-frequency acoustic systems. These ultrasonic calls travel through the air until they encounter objects, at which point they reflect back to the bat's highly specialized ears. The dynamic range of bat hearing is extraordinary, spanning from near silence at 0 decibel sound pressure level up to 140 decibels, the intensity of the bat's own calls at close range. This 140 decibel range, equivalent to hearing everything from a whisper to a jet engine, allows bats to process both the faint echoes from distant targets and the intense reverberations from nearby surfaces, without saturating their auditory system. The time delay between emission and echo return allows the bat to calculate distance with remarkable accuracy. Temporal resolution in some species reaches 400 nanoseconds, with certain species possibly achieving range resolution as fine as 10 nanoseconds. These figures translate to distance discrimination approaching 1/10 of a millimeter. The frequency content and intensity of returning echoes provide information about target size, shape, texture, and even material composition. Differences in timing and intensity between the 2 ears enable horizontal localization, accurate to within 1 to 2 degrees, while specialized ear structures, including the tragus, contribute to vertical positioning. The production of these calls requires specialized anatomy operating at extraordinary speeds. The larynx contains fast muscles capable of contracting at rates exceeding 200 times per second, far faster than muscles in most mammals can achieve. To prevent self-deafening from their own 140 decibel vocalizations, bats have evolved a remarkable mechanism. The middle ear bones disconnect during call production, physically uncoupling the sound transmission pathway and protecting the inner ear from damage. This disconnection and reconnection occurs with each call cycle, a feat of neuromuscular control synchronized with sub-millisecond precision. This sensory capability fundamentally reshapes what constitutes available habitat and prey. Diurnal raptors require sufficient light to visually detect and track flying insects or small vertebrates. Even birds with exceptional night vision, such as nightjars and owls, rely on ambient moonlight or starlight reflecting from prey items. Complete darkness renders these hunters effectively blind. Echolocation, by contrast, generates its own signal and requires no external illumination. A bat hunting insects on a moonless night in dense forest canopy operates with sensory clarity that no vision-dependent predator could achieve under the same conditions. The evolutionary radiation of bats into diverse ecological niches demonstrates how this sensory innovation opened previously inaccessible opportunities. Consider the big brown bat, a common North American species that hunts beetles and other large insects in open airspace. During the search phase of hunting, this bat emits calls with relatively shallow frequency modulation at a repetition rate of 5 to 10 Hz, with each call lasting 15 to 20 milliseconds. These search calls travel substantial distances, creating a sensory volume that can exceed 20 cubic meters, allowing the bat to detect potential prey at ranges exceeding 10 meters. The duty cycle during this phase, the percentage of time spent producing calls versus listening for echoes, remains below 10%, typical of frequency-modulated bats. Once a target is detected, the bat's behavior shifts dramatically. It transitions into an approach phase, increasing call repetition rate to 20 to 80 Hz while shortening call duration to 2 to 5 milliseconds and steepening the frequency modulation. This provides rapid updates on target position and velocity. In the final capture phase, sometimes called the terminal buzz, call duration drops to just half a millisecond, while repetition rate surges above 200 Hz. The bat is now receiving positional updates every 5 milliseconds, allowing it to track even evasive prey performing rapid aerial maneuvers. This three-phase hunting sequence, documented across dozens of species, reveals a sophisticated strategy of trading spatial coverage for temporal resolution and positional accuracy as the hunt progresses. The sensory volume concept illustrates a key advantage bats possess over visual hunters. During the search phase, a bat's echolocation creates a detection zone whose size can be dynamically adjusted. When hunting in open space, some species generate sensory volumes approaching or exceeding 1,000 cubic meters for large targets. As they approach clutter such as vegetation, they reduce this volume to avoid being overwhelmed by echoes from background objects. This adaptive control over sensory sampling has no direct parallel in vision-based hunting. A hawk scanning for prey from a perch has a fixed relationship between acuity and field of view determined by eye anatomy. A bat, by contrast, actively controls how much space it samples and at what resolution, adjusting these parameters dozens of times per second based on task demands and environmental conditions. The ability to operate in complete darkness extends the temporal niche available for foraging. Many flying insects emerge at dusk or during night hours, driven by their own adaptations to avoid diurnal predators. These insects represent a massive food resource that remains largely unexploited by vision-dependent hunters. Bats have diversified to exploit this resource across a remarkable range of scales and hunting strategies. The bumblebee bat, weighing about 2 grams and measuring just 3 centimeters in body length, represents one of the smallest mammals on Earth. At the opposite extreme, the spectral bat can have a wingspan exceeding 1 meter and hunts vertebrate prey, including birds, rodents, and other bat species. The auditory system of echolocating bats shows corresponding specializations. The inferior colliculus, a midbrain structure that processes sound, is proportionally larger than in most mammals and contains neurons tuned to detect specific features of echoes. Some neurons respond preferentially to echo delays in specific ranges, effectively creating a neural map of target distance. Others show sensitivity to Doppler shifts, the frequency changes caused by relative motion between bat and target. Still others respond to particular combinations of call and echo features, suggesting they extract information about target identity or behavioral state. Different bat lineages have evolved distinct echolocation strategies suited to their ecological niches. Species that hunt in open airspace often use calls with relatively narrow frequency bandwidths and longer duration, maximizing detection range at the cost of reduced ranging precision. Species hunting near vegetation use very short, broadband frequency-modulated calls that provide excellent range resolution and minimize the temporal overlap between target echoes and clutter echoes. Horseshoe bats and some other species have evolved a fundamentally different approach, using long constant frequency calls that allow them to detect flying insects through Doppler shift analysis. These constant frequency bats maintain duty cycles exceeding 30%, spending far more time producing sound than their frequency-modulated relatives. When a bat using this strategy flies toward a fluttering moth, the motion of both bat and insect wings creates complex Doppler shifts in the echo. The bat's auditory system is specifically tuned to detect these modulations, which reveal not just the presence of prey, but details about wing beat frequency and potentially even species identity. The behavioral flexibility of bat echolocation extends beyond basic call parameter adjustment. Some species actively control the directional aim of their sonar beam, moving their heads to scan the environment or focus on specific targets. This gaze control operates on timescales of tens of milliseconds, comparable to eye movement speeds in many vertebrates. When approaching prey in the presence of obstacles, bats can rapidly alternate their sonar beam between target and background, effectively time-sharing their attention between navigation and tracking. The greater mouse-eared bat demonstrates this when hunting near vegetation, shortening call duration to create windows in which echoes from the prey arrive without overlap from plant echoes, then adjusting both call timing and duration to maintain this separation as distances change during the approach. The sensory-motor integration required for successful echolocation hunting approaches exceeds the complexity of visual hunting in many respects. A bat pursuing an evasive insect must predict prey trajectory, plan an interception path, monitor and avoid obstacles, and continuously update its motor plan based on sensory feedback. The entire sequence from prey detection to capture often occurs in less than 1 second and requires processing hundreds of echolocation calls and their associated echoes. Recent research using onboard recording devices attached to free-flying bats reveals that they achieve this through a combination of stereotyped behavioral sequences and rapid adaptive responses. When pursuing prey in open space, the big brown bat maintains a remarkably consistent relationship between its flight velocity and the rate of sensory sampling. As it accelerates toward the target, it proportionally increases call repetition rate, maintaining approximately constant spatial redundancy in its sensory coverage. This allows the bat to obtain multiple independent echo measurements from each region of space it traverses, improving detection reliability and enabling it to track targets despite the noise and variability inherent in any sensory system. In the final moments before capture, redundancy increases further as call rate surges while flight speed decreases, providing the dense sensory information needed to predict the exact position where prey will be in the final wing beat before contact. The adaptive control extends to call intensity. Wild bats hunting in open conditions emit calls exceeding 130 decibels, creating sensory volume sufficient to detect small insects at ranges of 10 meters or more. As they close on prey, they reduce call intensity by 40 decibels or more, shrinking sensory volume and reducing the absolute intensity of returning echoes. This might seem counterproductive, but it serves multiple functions. It prevents saturation of the auditory system by overly intense echoes at close range. It reduces acoustic clutter by limiting the volume of space ensonified. And perhaps most significantly, it creates prey echoes that occupy a specific range of intensities to which the bat's auditory system is optimally tuned. Rather than passively receiving whatever echoes result from the environment, the bat actively regulates the strength of sensory input to match the processing capabilities of its nervous system. This active sensory control has implications for understanding how bats exploit ecological niches. A diurnal raptor hunting over a meadow faces a fixed relationship between lighting conditions, prey contrast, and detection range. The hawk cannot adjust the intensity of sunlight or the spectral properties of illumination. It must work with whatever visual information the environment provides. A bat, by contrast, has far greater control over the sensory information it receives. By adjusting call intensity, duration, bandwidth, and repetition rate, it can effectively tune its sensory system to match current task demands and environmental conditions. This flexibility becomes particularly important when considering the challenges of hunting in cluttered environments. Dense vegetation returns cascades of echoes from leaves, branches, and trunks, creating acoustic scenes of extraordinary complexity. Each echolocation call may generate dozens or hundreds of echoes, arriving at different delays from objects distributed across three-dimensional space. Extracting information about a single flying insect from this cacophony represents a formidable computational challenge. Bats solve this problem through multiple complementary strategies. They reduce call duration to minimize temporal overlap between echoes from different distances. They use broadband calls that provide better range resolution, allowing them to separate closely spaced objects. They adjust call repetition rate to maximize the number of echo-free gaps in which prey echoes can be received without interference. They alter flight speed and trajectory to change the spatial relationship between themselves, prey, and background clutter. And they exploit the directional properties of both their sonar emission and echo reception to emphasize on-axis targets while suppressing off-axis clutter. The directional properties of bat sonar deserve particular attention. While bats do not have the highly directional, tightly focused beams of engineered sonar systems, their calls and ears do show significant directionality. Emission patterns vary among species, but typically show a main beam width of 30 to 60 degrees with substantially reduced intensity off-axis. Reception patterns determined by ear shape and size show similar directionality. The combination of directed emission and directed reception creates what is effectively a flashlight beam of sound. Objects directly ahead of the bat return echoes that have been transmitted at high intensity and received with high sensitivity. Objects to the side experience both reduced emission intensity and reduced reception sensitivity, making their echoes substantially weaker even if they are at the same distance. This directionality provides a mechanism for perceptual segmentation of complex acoustic scenes. By aiming the sonar beam at a target of interest, the bat can enhance that target's echo relative to clutter echoes from other directions. This auditory foveation creates a graded attentional field similar in some respects to the center-surround organization of visual attention. Objects within the attended region receive preferential processing, while peripheral information is suppressed, though not entirely eliminated. The importance of this capability becomes clear when considering specific hunting scenarios. The gleaning bat Megaderma lyra hunts for prey on the ground or vegetation surfaces rather than in flight. It often detects potential prey by listening for sounds produced by prey movement, effectively using passive acoustic cues rather than or in addition to echolocation. Once it has localized a potential prey item, it must approach while navigating through three-dimensional vegetation structure, then precisely localize the stationary target immediately before attack. Throughout this sequence, the bat adjusts both its echolocation behavior and flight trajectory to maintain sensory contact with the target while avoiding obstacles. Video recordings of these hunts reveal complex coordination between gaze, flight path, and echolocation parameters. The bat initially homes in on prey-generated sounds, using passive hearing to obtain a coarse localization. As it approaches, it begins producing echolocation calls and uses the returning echoes to refine target position and begin assessing the local environment for obstacles. In the final approach, it may hover briefly while directing its sonar beam at the prey, obtaining high-resolution information about exact position and potentially about prey identity through echoacoustic features. The terminal attack combines information from multiple sensory modalities, using vision, echolocation, and passive acoustic localization to guide the final pounce. This multimodal integration characterizes much of bat sensory ecology, but is often overlooked in discussions focused narrowly on echolocation. Most bats have functional vision, and while their visual acuity is generally lower than that of similar-sized diurnal mammals, it provides useful information about large-scale environmental features and can aid in prey detection under favorable conditions. Some fruit-eating bats have excellent vision and use visual cues more than echolocation for finding food. Others combine visual and acoustic information in more balanced proportions. The integration of multiple sensory streams raises questions about how bats represent their environment. When a bat flies through a forest, it receives visual information about vegetation structure, echolocation echoes from nearby surfaces and objects, passive acoustic cues from environmental sounds and potential prey, mechanosensory input from air currents detected by specialized hairs on wings and body, and vestibular signals encoding head rotation and linear acceleration. These diverse information streams must be combined into a coherent spatial representation that guides navigation and hunting behavior. Current evidence suggests that bat spatial cognition shares fundamental features with that of other mammals despite the unusual importance of echolocation. Studies of hippocampal place cells in echolocating bats find neurons that respond when the animal occupies specific locations in space, similar to place cells in rats and other species. These cells integrate information across sensory modalities, responding to location regardless of whether the bat arrives there through vision, echolocation, or path integration based on self-motion cues. This suggests that echolocation feeds into the same general-purpose spatial mapping systems found throughout mammals rather than creating a separate acoustic spatial representation. The evolutionary success of bats in nocturnal predation niches reflects not just the raw capability of echolocation as a sensory system, but the sophisticated behavioral strategies that bats have evolved to exploit this capability. The three-phase hunting sequence, the dynamic adjustment of call parameters, the coordination between sensory sampling and flight motor control, and the integration of echolocation with other sensory modalities collectively create a hunting system that rivals the effectiveness of visual predation in equivalent diurnal niches. This effectiveness is quantifiable. Insectivorous bats can capture prey at rates exceeding one insect every few seconds during peak foraging periods. A colony of big brown bats may consume several thousand insects per night, substantially impacting local insect populations. Economic analyses estimate that insect consumption by bats provides billions of dollars in agricultural pest control services annually. This level of predation pressure is comparable to that exerted by diurnal insectivorous birds in the same habitats, despite the fundamental difference in primary sensory modality. The question then becomes what specific features of nocturnal environments and prey characteristics drove the evolution of such extreme sensory capabilities, and whether the physical constraints of echolocation impose limits on how far this sensory radiation can extend. Fish swimming below water surfaces present acoustic challenges that seem insurmountable. Blood flowing within sealed circulatory systems appears beyond acoustic detection. Yet bats have found pathways into these niches as well, suggesting that the interplay between constraint and innovation continues to generate solutions that expand what seemed to be fixed boundaries. Chapter 2. The Nocturnal Raptor. In the tropical forests of Central and South America, a creature hunts that defies easy categorization within traditional ecological frameworks. With a wingspan approaching 1 meter and weighing up to 190 grams, Vampirum spectrum represents something unprecedented in the mammalian world. a flying apex predator that occupies the same functional niche as owls and small raptors, yet operates through fundamentally different sensory and behavioral mechanisms. This is the spectral bat, the largest carnivorous bat species on Earth, and its existence challenges assumptions about the limits of bat ecological radiation. Most bat species consume insects or fruit. Bats have diversified into numerous feeding strategies, but the vast majority remain committed to relatively small prey items or plant materials. Only 9 bat species qualify as true carnivores, defined as regularly consuming vertebrate prey. Among these, the spectral bat stands alone in terms of size, hunting capability, and the consistent targeting of prey that in other ecosystems would fall exclusively to avian predators. The spectral bat's morphology reveals adaptations that parallel those of raptorial birds while maintaining the fundamental mammalian body plan. The skull is narrow and elongated with a pronounced sagittal crest, providing attachment surfaces for powerful jaw muscles. The overall structure has been compared to a miniature canine or bear skull, though the bat weighs a fraction of what those mammals do. The dental formula includes well-developed upper canine teeth and molars with reduced crushing surfaces relative to sharing surfaces, optimized for processing meat rather than insects or fruit. The bite force relative to body size exceeds that of any carnivorous mammal. Researchers estimate the spectral bat can deliver bites of 80 to 100 newtons, sufficient to crush the skull of prey animals approaching or exceeding the bat's own body mass. The wings show adaptations distinct from those of aerial insectivores. Rather than the long, narrow wings favored by bats that chase flying insects in open airspace, the spectral bat has relatively short, broad wings, creating a large surface area. The wingtips are rounded and almost squarish rather than pointed. This configuration produces exceptionally low wing loading of 20.05, the lowest body mass to wing area ratio among 51 neotropical bat species studied. The spectral bat cannot match the straight line velocity of bats specialized for open-air pursuit, but it excels at hovering, slow flight, and carrying heavy loads. These are precisely the flight characteristics needed for a predator that plucks sleeping birds from branches or captures rodents on the ground. Each thumb bears a large, recurved claw grooved in a manner similar to cat claws. These are the primary weapons for securing prey during the initial attack. When the bat strikes, it hooks these claws into the target, preventing escape while positioning itself to deliver the killing bite. The legs are long for a bat, and the feet are composed of slender bones, each digit equipped with well-developed claws. The combination creates a grasping apparatus that can hold struggling prey while the bat maneuvers for the optimal bite placement. The hunting strategy of the spectral bat shows remarkable convergence with owl predation, despite the different sensory modalities employed. Both predators hunt primarily at night. Both target vertebrate prey in the size range of small birds and rodents. Both use stealth approaches, minimizing detection by prey. Both kill with powerful bites delivered to the head or neck. Yet the sensory information guiding these parallel behaviors differs fundamentally. Owls rely primarily on vision, supplemented by acute hearing. The spectral bat, in contrast, uses echolocation for navigation, but appears to rely heavily on olfaction for prey detection. Multiple lines of evidence point to scent as the primary modality for finding prey. The spectral bat preferentially targets bird species known to have strong body odors. Cuckoos, trogons, and motmots all appear in the diet more frequently than would be expected based on their abundance alone. The groove-billed ani, which both produces a strong smell and roosts in groups, constitutes a particularly common prey item. The brain structure supports the importance of olfactory hunting. The spectral bat has well-developed olfactory bulbs, larger relative to brain size than those of most insectivorous bats. The overall brain-to-body mass ratio of 1 to 67 exceeds that of cats and dogs. The cerebral hemispheres show extensive convolution, and the corpus callosum connecting the two hemispheres is described as exceptionally thick. These features suggest sophisticated sensory integration and processing capability beyond what would be required for simple echolocation-based insect hunting. The actual hunting sequence combines multiple sensory inputs in a coordinated behavioral program. The bat emerges from its roost at dusk and begins patrolling. Flight is slow and low, typically within a few meters of the ground or moving through the understory beneath the forest canopy. When a potential prey item is detected, the bat adjusts its flight path to approach. As it nears the target, it may hover briefly, using its agile and maneuverable wings to maintain position while assessing the exact location. The final strike happens rapidly. The bat swoops down from above if attacking a bird on a branch, or pounces from a low approach if targeting prey on the ground. The thumb claws strike first, hooking into the target. Immediately following, the bat delivers a powerful bite directed at the skull. The combination of crushing force and sharp canine teeth typically penetrates the cranium, causing immediate death or incapacitation. The size of prey taken reflects the bat's substantial capabilities. Individual prey items range from 20 grams for small birds up to 150 grams for the largest documented prey, the white-tipped dove. At the upper end of this range, prey weighs nearly as much as the predator itself. The bat's low wing loading becomes critical here, allowing it to lift off while carrying such heavy loads. After a successful strike, the bat typically carries the prey back to its roost rather than consuming it at the capture site. The remains reveal selective consumption patterns. The spectral bat discards bird feathers, bat wings from captured congeneras, and rodent tails. It consumes the muscle, organs, and often crushes bones to access marrow. The caloric density of vertebrate prey means the bat may need to feed only once every 2 to 3 nights during periods when large prey items are successfully captured. The spectral bat also consumes other bat species, creating a situation where a mammal preys upon other mammals in the same ecological guild. Documented prey includes the Highland yellow-shouldered bat, Geoffroy's tailless bat, Pallas's long-tongued bat, various short-tailed fruit bats, and even the common vampire bat. The social structure of spectral bats differs markedly from both the large colonies characteristic of many bat species and the solitary habits of most raptors. The spectral bat is monogamous, a rare trait among bats. Colonies consist of an adult male and female and their offspring. The typical roost contains 1 to 5 individuals. This small group size reflects the resource requirements of carnivory. A large colony of vertebrate-eating predators would rapidly deplete local prey populations. Radio tracking studies reveal home ranges of approximately 3.2 hectares, and the low density of spectral bats across their range, with wide spacing between roost sites, prevents overexploitation of prey resources. The family structure supports an unusual degree of parental investment. The adult male brings food back to the roost to provision both the adult female and their offspring. This biparental care is extremely rare in bats. In most species, males contribute nothing beyond sperm, and females raise young without assistance. The spectral bat male actively participates in feeding the family. He will carry prey items back to the roost, allowing the female to remain with dependent young while still obtaining adequate nutrition. This provisioning extends beyond the nursing period, continuing while young bats learn to hunt for themselves. The extended period of parental care allows young bats to develop the sophisticated hunting skills required for capturing vertebrate prey. Wild spectral bats can live at least 8 years, based on observations of individual bats returning to the same cave roosts, though the average lifespan remains unknown. This relatively long lifespan for a bat of this size reflects the reduced mortality risk associated with being an apex predator rather than prey, though it also means slow population recovery from disturbances. The geographic range extends from southern Mexico through Central America and into northern South America, reaching as far south as central Bolivia and Brazil. Within this range, the spectral bat associates strongly with tropical rainforests, though it has been documented in the Cerrado savanna of Brazil and occasionally in pastures and orchards near forest edges. The elevation range extends from sea level to 1,650 meters, though in Mexico the species is restricted to lowland areas below 150 meters. This distributional pattern places the spectral bat in direct ecological overlap with multiple owl species and small raptors that hunt similar prey. The spectral bat hunts almost exclusively at night, emerging at dusk and returning to roost before dawn. While some owls are active during this same period, many small raptors that might compete for rodent or bird prey are strictly diurnal. This temporal separation reduces direct competition. Even among nocturnal predators, the spectral bat's use of scent for prey detection may provide access to prey that owls would miss. A bird roosting in dense foliage might be invisible and silent, escaping detection by visual or acoustic hunting, but its scent plume could betray its location to an olfactory predator. The bat's flight capabilities in cluttered environments exceed those of most owls. The low wing loading and highly maneuverable flight allow navigation through dense understory vegetation that would challenge even specialized forest owls. Perhaps most significantly, the spectral bat can access vertical microhabitats in ways that terrestrial or perch hunting predators cannot. A small mammal running along a branch 30 meters above the ground occupies a niche largely inaccessible to ground-based predators and difficult for perch hunting owls to exploit. The bat can approach from below, hover beneath the branch, and strike upward, using three-dimensional mobility that owls, despite their flight capability, rarely employ in actual hunting. The spectral bat's closest living relative is the big-eared woolly bat, Crotopterus auritus, which also consumes vertebrate prey, though not as consistently or exclusively. The two genera Vampirum and Crotopterus diverged approximately 14 million years ago, both descending from an insectivorous ancestor. This time frame suggests that the carnivorous specialization is relatively recent in evolutionary terms, though still ancient enough to have produced sophisticated adaptations. The spectral bat is not alone in filling raptor-like niches. The ghost bat of Australia, Macroderma gigas, has independently evolved a similar lifestyle, hunting vertebrate prey including birds, reptiles, and small mammals. This convergent evolution across continents in distantly related bat lineages suggests that the nocturnal vertebrate predator niche represents a genuine ecological opportunity accessible to bats given appropriate adaptations. What remains unclear is why more bat lineages have not pursued this strategy. This may reflect the difficulty of the transition. Insect hunting requires detecting, tracking, and capturing small, evasive targets, but the individual prey items have limited capacity to fight back or inflict injury. Vertebrate prey, even small birds or rodents, have defensive capabilities, including beaks, teeth, and claws that can injure an attacker. The morphological changes required for successful vertebrate predation, particularly the reinforced skull and powerful bite force, may represent a significant evolutionary hurdle that few lineages have crossed. The conservation status of the spectral bat reflects the vulnerabilities inherent in apex predator ecology. The International Union for Conservation of Nature lists the species as near threatened globally, though it has been classified as endangered in Bolivia and Mexico since 2001. The species faces pressure from habitat destruction and its naturally low population density. As a top predator with large home ranges and slow reproductive rate, producing only one pup per year, the spectral bat cannot quickly recover from population declines. The loss of primary forest habitat reduces both roost site availability and prey abundance. Yet the spectral bat persists across much of its historical range, a testament to the effectiveness of its adaptations. The morphological specializations for power and maneuverability, the sophisticated sensory integration combining olfaction with echolocation, the complex social structure enabling extended parental care, and the behavioral flexibility to exploit diverse prey types all work together to create a predator that fills a niche textbooks assign to birds. The question that emerges is not simply how bats achieve this, but whether similar unexplored opportunities exist elsewhere in their ecological radiation, waiting for the right combination of selection pressures and evolutionary innovations to unlock them. Chapter 3. Fishing Bats. The water surface presents a paradox for echolocating mammals. Sound waves traveling through air strike the interface between air and water and largely reflect back, with minimal penetration into the aquatic realm. The acoustic impedance mismatch between these two media creates an almost perfect mirror for ultrasonic frequencies. This physical reality would seem to place a hard boundary on where bats can hunt, confining them to the aerial domain and leaving aquatic prey to visual predators like kingfishers, herons, and ospreys. Yet several bat lineages have crossed this boundary, developing hunting strategies that allow them to extract fish and aquatic invertebrates from ponds, rivers, and coastal waters despite this fundamental limitation. The evolutionary pathway toward piscivorian bats likely began with trawling, a hunting technique in which bats fly low over water surfaces and capture insects floating on or emerging from the water. Many insectivorous bat species occasionally trawl, dragging their feet through the water to snag prey. The transition from trawling for insects to actively fishing for vertebrate prey required solving multiple challenges simultaneously. Detecting submerged or partially submerged fish despite the acoustic barrier, developing the physical capability to capture and lift struggling prey that can substantially exceed the mass of typical insect targets, and managing the energetic costs of hunting prey that may be more dispersed and less predictable than insect swarms. The greater bulldog bat, Noctilio leperinus, represents the most specialized fishing bat and demonstrates how far this adaptation can be pushed. Found from Mexico through Central America and into northern Argentina, this species has undergone morphological changes so extensive that it bears little superficial resemblance to its insectivorous relatives. The feet are the most immediately striking feature. They are enormous relative to body size, with toes spreading to create a grasping apparatus that can span 8 centimeters. Each toe terminates in a sharp, laterally compressed claw that curves into a hook shape. The claws themselves reach lengths of up to 2 centimeters in some individuals. When the bat strikes the water, these feet function like gaffing hooks, penetrating fish scales and securing prey that might otherwise slip free. The legs supporting these feet show corresponding specialization. They are longer than in typical bats, positioning the feet well below the body during flight. The hind limb musculature is robust, providing the power needed to drive the claws through fish skin and then lift the captured prey from the water. The calcar, a cartilaginous spur extending from the ankle that normally supports the tail membrane in bats, is particularly well developed and helps maintain tension in the membrane between the legs. This uropatagium is not merely structural. During fishing strikes, it functions as a scoop or net, creating a pouch that can cradle fish as the bat's claws secure them. The wings themselves differ from those of aerial insectivores in ways that parallel the wing adaptations seen in the spectral bat, but serve different functions. Where the spectral bat has broad wings for maneuverability and load carrying in cluttered environments, the greater bulldog bat has long, narrow wings that generate efficient sustained flight. The aspect ratio is high, similar to that of seabirds adapted for long-distance flight over water. This configuration allows the bat to patrol stretches of river or coastline for extended periods while expending minimal energy. The wing membrane extends to the ankle rather than the toe, leaving the feet free for prey capture without interference from the flight surfaces. Fish swimming near the surface create characteristic disturbances that betray their presence to an acoustic predator. Ripples from fins breaking the surface, turbulence from body movement just below the interface, and in some cases, the insects or small crustaceans that the fish themselves are pursuing. These disturbances create texture on the water surface that differs detectably from smooth, undisturbed water. Research using controlled experiments reveals the precision of this detection capability. When researchers presented bats with smooth metal plates versus textured plates designed to mimic the appearance of ripples, the bats preferentially investigated the textured targets. They can discriminate between ripples as small as 2 to 3 millimeters in amplitude. The bat flies low, typically within 20 to 50 centimeters above the water surface, adjusting its echolocation to detect these surface disruptions. When a disturbance is detected and the bat closes on the target, the feet enter the water at an angle, carving through the surface rather than slapping down flat. This reduces splash and maintains flight stability. The depth of foot insertion varies with target behavior. Sometimes only the toes penetrate the water, while in other strikes the foot may submerge to the ankle. The claws hook into the fish's body, and the entire foot contact with water lasts only a fraction of a second. The bat's momentum carries it forward and upward, and the powerful hind limb muscles retract the feet, pulling the fish from the water. This hunting method succeeds frequently enough to support the species across its extensive range. Fishing bats can capture multiple fish per hour during productive foraging bouts. The prey includes small fish typically ranging from 3 to 10 centimeters in length, though occasional captures of larger individuals up to 15 centimeters have been documented. The fish species taken reflect what is locally abundant and accessible near the surface. In marine environments, bulldog bats capture small schooling fish. In rivers and lakes, they take various minnows, killifish, and juvenile fish of larger species. The convergence with piscivorous birds becomes apparent when comparing hunting strategies. The belted kingfisher, common across North America, hunts from perches overlooking water. It watches for fish moving below the surface, then dives head first to capture prey with its sharp beak. The hunting osprey circles above water, spots fish from heights of 10 to 40 meters, then plunges feet first to grasp prey with sharp, curved talons. The fishing bat uses the feet-first strike strategy of the osprey, but operates from low altitude, like a surface-feeding bird. All three predators converge on solving the same fundamental problem, extracting fish from water where the predator itself cannot survive immersion for more than brief moments. The osprey's talons show remarkable morphological convergence with the fishing bat's claws. Both are sharply curved, both have rough surfaces that improve grip on slippery prey, and both are powered by strong flexor muscles that can maintain crushing force even as prey struggles. The osprey has additionally evolved reversible outer toes that can be positioned to provide a 2 in front, 2 in back grip configuration, something the bat cannot achieve with its fixed mammalian digit arrangement. Yet the bat compensates through the use of the uropatagium as a secondary capturing surface and through behavioral flexibility in strike angle and foot placement. The kingfisher's hunting approach differs more from the bat's strategy, relying on visual detection and a head-first plunge. Yet the ecological result is similar. Both predators exploit small fish in shallow water that larger piscivores would struggle to access efficiently. The kingfisher's small size allows it to hunt in narrow streams and small ponds, where herons or ospreys could not effectively forage. The fishing bat similarly specializes in environments where its low-altitude flight and echolocation-based detection provide advantages over visual hunters operating from perches or from high-altitude searches. The sensory basis for prey detection represents the clearest distinction between bat and bird piscivores. Birds rely almost entirely on vision. They watch for movement, for the flash of scales, for the distortion of the water surface that indicates a fish just below. This visual hunting requires adequate light. Kingfishers and ospreys are strictly diurnal. They become ineffective as light fades. The fishing bat operates in complete darkness. It hunts during the hours when visual predators have ceased activity. This temporal separation reduces direct competition and provides access to fish that are active at night or that rise to the surface in darkness to feed on nocturnal insect hatches. The fishing bat's echolocation-based hunting does face constraints that visual predators avoid. Turbulent water obscures the subtle ripples that indicate fish presence. Heavy rain creates so many surface disturbances that the signal-to-noise ratio for fish detection drops to unusable levels. Wind-driven waves similarly interfere with detection. Fishing bats show strong preferences for calm conditions and protected waters where surface texture remains informative. This restricts their foraging to particular times and locations. An osprey can hunt successfully even in moderate wind and wave conditions because it sees fish beneath the surface rather than relying on surface disturbances. Yet the acoustic modality provides advantages in other contexts. Fishing bats can hunt in areas of complete darkness where even moonlight does not penetrate, such as in caves with water-filled passages or in dense forest canopy overhanging streams. They can detect fish in murky water where visibility would be near zero for a visual predator. The ripples and surface disturbances that reveal fish presence occur regardless of water clarity. This potentially expands the hunting grounds available to fishing bats into niches that birds cannot exploit. The evolutionary transition from insectivory to piscivory occurred independently in multiple bat lineages, though not all have progressed to the same degree of specialization as the greater bulldog bat. Several species in the genus Myotis, particularly the long-fingered bat Myotis capucini found around the Mediterranean and Daubenton's bat Myotis daubentonii of northern Europe, regularly capture small fish while also continuing to take insects. These species show intermediate levels of morphological specialization. Their feet are larger than those of strictly insectivorous relatives, but not approaching the extreme development seen in bulldog bats. They hunt by flying low over water, detecting surface disturbances, and raking their feet through the water in areas where fish activity is detected. The success rates appear lower than those of specialized fishing bats, but the strategy provides supplemental nutrition during periods when fish are abundant near the surface. Myotis capucini, found in freshwater and coastal habitats around the Mediterranean, feeds extensively on small fish during warm months when fish congregate in shallow waters. It has been observed capturing fish from both still and flowing waters, adjusting its flight patterns to the specific hydrological conditions. Myotis daubentonii show similar flexibility, hunting insects over water during insect emergence periods and shifting to fish when surface-feeding fish are detectable. Both species retain the ability to forage successfully on insects alone, suggesting they have not crossed the threshold into obligate piscivory that would require the more extreme morphological specializations seen in Noctilio leperinus. This raises questions about the trajectory of evolution toward increasing piscivorous specialization. Does the fishing adaptation proceed through a long period of facultative piscivory in which bats opportunistically take fish when available while maintaining insectivory as the primary feeding mode? Or do some lineages transition rapidly once key morphological or behavioral innovations arise? The phylogenetic distribution of fishing behaviors suggests both pathways may occur. The bulldog bats represent an entire genus, Noctilio, with two species. The lesser bulldog bat, Noctilio albiventris, feeds primarily on insects, particularly those on or emerging from water surfaces, but occasionally captures small fish. This pattern suggests the genus initially evolved trawling behavior for insects, then the greater bulldog bat lineage pushed further into piscivory, while the lesser species retained a more generalized diet. The two species are similar in size and often found in the same geographic regions, sometimes roosting in mixed groups. Yet the lesser bulldog bat's feet are large compared to purely aerial insectivores, but smaller and less specialized than those of its piscivorous congener. When insects are abundant, the lesser species fills its stomach with beetles, moths, and other large insects. During periods when insect availability drops, it has been observed to capture occasional fish, demonstrating behavioral flexibility but not the committed piscivory of its congener. The energetic calculus of piscivory differs from that of insectivory in ways that influence which bat species can successfully make the transition. Insects are small, numerous, and can be captured in rapid succession during productive foraging bouts. A bat can consume hundreds of insects in a few hours, accumulating the necessary energy intake through many small captures. Fish are individually larger and more energy-dense, but they are caught less frequently. A fishing bat might capture 10 to 30 fish in a night of hunting rather than hundreds of insects. The handling time per prey item increases. The fish must be carried to a perch or roost, positioned properly in the mouth, and consumed. This takes longer than the in-flight consumption of small insects. For this shift to be viable, the energy gained per fish must be sufficient to justify the reduced capture rate. This tends to favor larger bat species making the transition to piscivory. A small bat with high mass-specific metabolic rate might struggle to meet its energy needs capturing fish at the rate fishing behavior allows. A larger bat with lower mass-specific metabolism can more easily afford the longer intervals between captures. The greater bulldog bat, with mass ranging from 40 to 90 grams depending on sex and condition, sits at a size range where piscivory becomes energetically feasible. It is substantially larger than most insectivorous Myotis species, and this may not be coincidental. The distribution of fishing bats concentrates in tropical and subtropical regions where fish productivity is high and water bodies support abundant small fish populations year-round. Temperate regions have fewer piscivorous bat species, and those that do fish, like the Myotis species mentioned earlier, often shift to primarily insectivorous diets during seasons when fish are less available or accessible. This geographic pattern mirrors the distribution of many piscivorous birds, which similarly show higher diversity and specialization in tropical waters. The question of how fishing bats locate productive fishing grounds highlights the role of learning and memory in foraging. Unlike insects that form predictable swarms at consistent locations and times, fish distributions can be more variable. A section of river that yields abundant prey one night might be nearly empty the next. Fishing bats could search randomly each night, but this would be energetically costly. Limited evidence from tracking studies suggests fishing bats do show sight fidelity to particular stretches of river or lakeshore, returning repeatedly to areas that have previously yielded successful hunts, while continuing to sample new areas. The sensory integration required for successful fishing extends beyond echolocation. Fishing bats retain functional vision, and while their visual acuity is modest compared to diurnal animals, they can detect large-scale features of the environment and potentially use visual cues to select foraging locations. The combination of learned knowledge about where fish are likely to be found, visual navigation to reach those locations, and echolocation to detect and capture individual fish creates a multimodal hunting system. The ability to take flight from the water surface represents another crucial adaptation. When a bat's feet and sometimes parts of its wing membrane contact water during a fishing strike, the added mass and drag could prove catastrophic if the bat cannot generate sufficient lift to pull itself free. Greater bulldog bats have been observed to swim using rowing motions of the wings if they become immersed. And they can take off directly from water by using rapid, powerful wing beats to accelerate to flight speed. This capability likely evolved as a safety mechanism, preventing drowning during failed strikes, but it expands the behavioral options available to the bat. A fishing bat can potentially rest briefly on the water surface between strikes or even swim short distances to retrieve prey that was struck but not immediately secured. These swimming and water takeoff abilities, while present in fishing bats, have not been documented to the same degree in most other bat species. Insectivorous bats that accidentally contact water while drinking or hunting often struggle to regain flight and may drown if they cannot quickly reach a shore where they can climb to a launch point. The evolutionary story of fishing bats reflects a broader pattern. Bat radiations have filled ecological niches typically occupied by very different animals. Piscivory evolved in birds repeatedly across multiple lineages, from kingfishers to ospreys to herons to cormorants, each developing distinct strategies for exploiting aquatic prey. Mammals, generally less suited to the aquatic interface due to their need to maintain body temperature and their respiratory requirements, have produced fewer piscivorous specialists. Yet bats, through their flight capability and echolocation, found a pathway into this niche.
They hunt where birds cannot, in complete darkness, and they do so with sensory mechanisms birds do not possess. The result is not a bat that becomes bird-like, but rather a bat that solves bird-like problems through mammalian and chiropteran-specific innovations. What other ecological opportunities remain accessible to bats, hidden within parameter spaces that current species have not yet explored?
Chapter 4. Mammalian Blood-Feeders. Blood feeding as an ecological strategy has evolved repeatedly across the animal kingdom, yet it remains predominantly the domain of arthropods. Mosquitoes, ticks, fleas, leeches, and various flies have independently discovered that vertebrate blood provides a nutrient-dense food source available in enormous quantities wherever warm-blooded animals congregate. Mammals, with rare exceptions, do not feed on blood. The challenges are substantial. Blood is mostly water and must be processed quickly to concentrate nutrients while avoiding fluid overload. It clots rapidly, requiring specialized anticoagulants, and obtaining it requires either parasitic attachment or repeated feeding events that expose the predator to host defenses. Yet 3 species of bats in the subfamily Desmodontinae have crossed this boundary, becoming the only mammals that feed exclusively on blood, and in doing so, occupying an ecological niche that textbooks assign to invertebrates. The common vampire bat, Desmodus rotundus, is the most widespread and best studied of these species. Found from northern Mexico through Central America and into northern Argentina and Chile, it feeds primarily on the blood of large mammals. In contemporary landscapes, this means predominantly domestic livestock, particularly cattle and horses, though it historically fed on native ungulates and other large mammals before the expansion of ranching provided abundant and predictable food sources. The white-winged vampire bat, Diamus youngi, and the hairy-legged vampire bat, Dipilla ecodata, are less common and feed primarily on birds, occasionally taking blood from mammals as well. All 3 species are relatively small, with body lengths of 7 to 9.5 centimeters and masses around 40 grams. The common vampire bat has a wingspan of 15 to 18 centimeters, modest even by the standards of insectivorous microbats. The physical appearance of vampire bats reveals adaptations that serve blood feeding while retaining the basic chiropteran body plan. The teeth represent perhaps the most specialized feature. The upper incisors are triangular with extremely sharp points and lack enamel. This absence of enamel might seem counterintuitive, as enamel provides hardness and resistance to wear in most mammals. In vampire bats, the lack of enamel serves a specific function. The teeth undergo thagosis, a process of self-sharpening in which the upper incisors continuously brush against the lower canines during jaw movements. The result is incisors that remain razor-sharp throughout the bat's life, capable of making precise cuts through skin with minimal pressure. The wound created is distinctive, a small crater-like incision, typically 7 millimeters wide and 8 millimeters deep, clean-edged and deep enough to access capillary beds while avoiding larger blood vessels that might bleed too profusely. The saliva of vampire bats contains a sophisticated cocktail of bioactive compounds that parallel the chemical arsenals of blood-feeding arthropods while showing independent evolutionary origins. Anticoagulants prevent the blood from clotting at the wound site. The most studied of these is a glycoprotein appropriately named Draculin, which inhibits factor X, a key component of the clotting cascade. This compound has attracted medical interest, and synthetic versions have been developed for treating stroke patients, where dissolving blood clots can prevent brain damage. Additional compounds in the saliva prevent vasoconstriction, keeping blood vessels at the wound site dilated to maintain flow. Others provide local anesthesia, reducing the likelihood that the host animal will wake or react to the feeding bat. The combined effect allows the bat to feed for 20 to 30 minutes on a sleeping host that remains unaware of the intrusion. This chemical warfare against host defenses shows remarkable convergence with the strategies of ticks, mosquitoes, and other blood-feeding arthropods. Female mosquitoes inject saliva containing anticoagulants, vasodilators, and immunomodulatory compounds. Ticks produce similar cocktails, often with even more complex mixtures because their attachment and feeding periods extend over days rather than minutes. Yet the molecular details differ. The specific proteins and compounds in vampire bat saliva are mammalian proteins that have been co-opted and modified for this novel function. They are not homologous to the arthropod compounds serving similar purposes. This represents convergent evolution at the biochemical level, independent lineages solving the same problems with different molecular tools. The detection of suitable hosts relies on sensory capabilities that differ fundamentally from those of arthropod blood feeders. Mosquitoes and ticks use carbon dioxide detection, body heat, and chemical cues from skin secretions to locate hosts. Vampire bats employ these same general principles but implement them through different anatomical structures. The bat's hearing is extraordinarily sensitive to the low-frequency rhythmic breathing sounds produced by sleeping mammals. The brain contains specialized neural circuits that can detect and recognize breathing patterns, distinguishing the regular respiration of a sleeping animal from the irregular breathing of one that is awake. This allows the bat to identify vulnerable hosts before even approaching. Heat detection occurs through specialized receptors on the nose. These thermoreceptors can detect infrared radiation emitted by warm-blooded prey, functioning analogously to the pit organs of some snakes. The bat uses this thermal sense to identify specific locations on the host where blood flows close to the skin surface. Areas with minimal fur or feather coverage, where major blood vessels run superficially, or where skin temperature is slightly elevated due to nearby vasculature, all become apparent to the bat's thermal vision. This guides the bat to optimal feeding sites, maximizing the blood flow it can access while minimizing the depth of incision required. The approach to the host and the feeding process itself reveal behaviors with no real parallel in arthropod blood feeders simply because bats are not parasites that attach to hosts for extended periods. The vampire bat lands near but not on the sleeping animal, approaching on foot. Here, the bat's then usual locomotor abilities become critical. Unlike most bat species, which are awkward when grounded, vampire bats can walk, hop, and run with considerable agility. They use a quadrupedal gait, but unlike the splayed limb crawling of many bats, vampire bats can support their weight on their hind limbs and use their modified thumbs as additional contact points. They can move rapidly across the ground, approaching the host stealthily. When ready to feed, the bat climbs onto the host, often positioning itself on a limb or the shoulder where a major blood vessel runs close to the surface. The sharp incisors make a quick, precise cut. The bat then laps blood from the wound, channeling it down a groove on the dorsal surface of the tongue. This lapping action differs from the sucking mechanism of mosquitoes or the passive feeding of attached ticks. The bat is actively consuming blood at a rapid rate, its tongue moving in quick succession to draw fluid into the mouth and down the throat. The digestive system must immediately address the challenge that blood is approximately 83% water with relatively low caloric density compared to solid food. A typical feeding session for a common vampire bat results in the consumption of 20 to 30 grams of blood, approaching or exceeding the bat's own unfed body weight. This enormous fluid load would be catastrophic if not managed. The stomach and intestine begin rapidly absorbing water from the blood meal within minutes of feeding. This water is shunted to the kidneys, which filter it and pass it to the bladder. Remarkably, vampire bats begin urinating within 2 minutes of starting to feed. Throughout the feeding session and immediately after, the bat voids large quantities of dilute urine, shedding the excess water while retaining the protein, lipid, and other nutrients in the blood. This rapid fluid processing represents a physiological feat without direct parallel in blood-feeding arthropods. Mosquitoes also consume blood meals that can double their weight, but they concentrate the meal more gradually and typically remain at or near the feeding site during the early stages of digestion.
Ticks, feeding over days, process the meal even more slowly. The vampire bat must manage its fluid load rapidly because it needs to fly shortly after feeding. Carrying an additional 20 to 30 grams of fluid while flying would be energetically prohibitive and mechanically difficult. The ability to begin jettisoning water even while still feeding allows the bat to take flight soon after completing the meal, returning to its roost before dawn.
The metabolic consequences of an exclusive blood diet create further challenges. Blood is rich in protein but contains virtually no carbohydrates and minimal fat. The bat must derive all its energy from protein metabolism, which is unusual for a mammal and creates specific biochemical demands. The breakdown of amino acids produces nitrogen waste in the form of urea, which must be excreted. The kidneys of vampire bats are adapted for processing high protein loads and producing concentrated urine to conserve water while eliminating nitrogen waste. The liver also shows specializations, with enhanced capacity for processing amino acids and synthesizing the compounds needed for normal metabolism from protein building blocks. Recent research has revealed an even more remarkable metabolic adaptation. Vampire bats can derive up to 60% of their energy from rapid amino acid breakdown during flight, with this conversion occurring within just 10 minutes of consuming blood. This metabolic feat is described as unparalleled in mammals and represents striking convergent evolution with blood-feeding insects, which similarly mobilize nutrients from blood meals with exceptional speed. The ability to rapidly convert ingested protein into flight fuel allows vampire bats to remain active and return to their roosts even while carrying the heavy fluid load of a fresh blood meal. Vampire bats have also lost the ability to synthesize certain enzymes that most mammals produce. They lack maltase, the enzyme that breaks down malt sugar, in their intestinal tract. This appears to be an evolutionary consequence of their specialized diet. Blood contains no maltose, so the selective pressure to maintain maltase production disappeared. The gene remained in the genome but accumulated mutations that rendered it non-functional. This kind of loss-of-function evolution is common when species become highly specialized, but it also constrains their future evolutionary options. A vampire bat lineage could not easily return to a frugivorous or insectivorous diet because it has lost genetic machinery that would be difficult to re-evolve. The social behavior of vampire bats shows sophistication that exceeds what is documented in blood-feeding arthropods and relates directly to the challenges of obligate hematophagy. Vampire bats can only survive 2 to 3 nights without feeding. This creates a precarious existence where a single unsuccessful hunting trip can lead to starvation. The bats have evolved a remarkable system of reciprocal food sharing to buffer against this risk. When a bat returns to the roost having failed to obtain a blood meal, it may beg from roostmates that fed successfully. The donor bat regurgitates a portion of its blood meal, transferring it mouth to the starving individual. This food sharing occurs both between related individuals and between unrelated roostmates, suggesting it operates through reciprocal altruism rather than purely kin selection. Research tracking individual bats over extended periods reveals that food-sharing relationships are not random. Bats preferentially share with individuals that have helped them in the past, creating a network of reciprocal relationships. Females in particular form long-term bonds with specific roostmates, and these bonds predict food-sharing patterns better than genetic relatedness. This represents a level of social cognition and memory uncommon in bats, and without parallel in blood-feeding arthropods. A tick does not share its blood meal. A mosquito cannot regurgitate to feed a hungry conspecific. The vampire bat's food sharing reflects both the precarious nature of blood feeding as a lifestyle and the cognitive and behavioral flexibility that being a mammal provides. The colony structure itself differs from the aggregations of most blood-feeding invertebrates. Vampire bats roost in groups ranging from a few individuals to several hundred in particularly favorable sites such as large caves or hollow trees. Within these roosts, social organization is complex. In common vampire bats, typical colonies contain several adult females and their offspring, along with one or more adult males. Some populations show a harem-like structure, where a dominant male mates with multiple females and defends them against other males. In other populations, males and females associate more loosely. In both cases, the long-term stability of female associations and the food-sharing networks among them form the social core of the colony. The reproduction and parental care patterns reflect the energetic constraints of hematophagy. Female vampire bats typically give birth to a single pup per year. The extended period of parental care, during which the pup depends on its mother for food, exceeds what is typical for bats of comparable size. Young vampire bats begin flying at about 3 months of age, but continue to receive food from their mother for several additional months as they learn to hunt. The mother regurgitates blood meals to feed the pup even after weaning from milk. Adult males in some populations also provision females and their young, bringing blood meals back to the roost to share. This extended parental investment and food provisioning is more typical of larger mammals or social carnivores than of bats, and it has no parallel in blood-feeding arthropods, which generally provide no parental care beyond egg placement. The locomotor specializations of vampire bats extend beyond their quadrupedal running ability. They are capable of leaping into the air from a standing position on the ground, achieving heights of up to 1.2 meters or 4 feet. This jumping ability serves a critical function. After feeding and processing enough fluid to make flight possible, the bat needs to become airborne. Most bats require a launching point, such as a branch or cliff edge, from which they can drop and gain speed before their wings generate sufficient lift. The vampire bat can launch directly from the ground or from a low perch on the host animal itself. The jump is powered primarily by the forelimbs, with the bat using its thumbs and wrists to push off explosively. Once airborne from the jump, the wings take over and the bat transitions to powered flight. This exceptional launching capability, rare among bats, directly supports the blood-feeding lifestyle by allowing rapid escape after feeding. This terrestrial agility and jumping capacity represent adaptations that facilitate blood feeding but also potentially open other ecological opportunities. Vampire bats are observed to feed on sleeping birds, approaching them on the ground or on low branches, making the incision and feeding while the bird remains asleep. They have been documented feeding on domestic chickens, wild ground-dwelling birds, and even seabirds on nesting colonies. The ability to move effectively on the ground and to take flight from ground level allows exploitation of hosts that would be difficult for a more typical bat to access. The geographic distribution of vampire bats is limited to the neotropics, from Mexico through northern Argentina. The northern limit appears to be constrained by temperature. Vampire bats cannot hibernate, and their metabolic requirements demand feeding every 2 to 3 nights. In regions where winter temperatures drop low enough to suppress host activity, or where livestock are kept indoors during cold months, vampire bats cannot persist. The southern limit of the range may be determined by similar factors, though habitat availability and historical biogeography also play roles. Within this range, vampire bats have benefited enormously from the introduction of European livestock following colonization. Cattle, horses, and pigs provided a massive increase in available food resources. Vampire bat populations expanded in response, and their ranges extended into areas where they had been absent or rare. This human-mediated range expansion created conflicts. Vampire bat bites can transmit rabies virus to livestock and occasionally to humans. The economic impact of vampire bat predation on livestock, both through direct blood loss and through disease transmission, has led to extensive vampire bat control programs involving the culling of colonies. These control efforts create an ironic situation where the expansion of livestock populations initially benefited vampire bats, but the subsequent human response threatens them. Culling programs using poison or physical destruction of roosts have reduced vampire bat populations in some areas. Paradoxically, disruption of colonies can temporarily increase rabies transmission rates by causing bats to disperse and contact new populations. The public health dimension of vampire bat ecology adds complexity to conservation considerations, though the bats themselves are not currently threatened with extinction across most of their range. The evolutionary origin of hematophagy in vampire bats remains somewhat unclear. The subfamily Desmodontinae is part of the larger family Philostomidae, the New World leaf-nosed bats, which includes species with highly diverse diets. Most philostomans are insectivorous, frugivorous, or nectarivorous. The closest relatives of vampire bats within this family are not blood feeders. This suggests hematophagy evolved relatively recently within the Desmodontinae lineage after it diverged from other philostomids. Molecular dating estimates place this divergence at roughly 20 to 30 million years ago, though the specific timing of the shift to blood feeding within the vampire bat lineage is uncertain.
One hypothesis suggests the transition occurred through an intermediate stage of feeding on blood from wounds on large mammals created by other predators or parasites. Large mammals in tropical forests experience various injuries from fighting, predation attempts, and heavy ectoparasite loads. Fresh wounds provide accessible blood without requiring specialized adaptations to create the wound. A bat that initially fed on insects or fruit might have begun opportunistically lapping blood from such wounds, gaining nutritional benefits that favored behavioral and morphological changes toward more dedicated blood feeding. Over time, this could have led to the evolution of the sharp incisors needed to create wounds, the saliva compounds needed to keep blood flowing, and the physiological adaptations for processing an all-blood diet. An alternative hypothesis suggests vampire bats evolved from ancestors that fed on ectoparasites of large mammals. Bats that landed on sleeping animals to consume ticks, flies, or lice might have occasionally bitten the host's skin while foraging, obtaining blood and discovering this resource. The progression from ectoparasite feeding to blood feeding would have involved gradual changes in behavior and morphology, potentially with an extended period where both resources were exploited.
Current evidence does not definitively resolve which pathway occurred, or whether some other scenario explains the origin of vampire bat hematophagy. What is clear is that once committed to blood feeding, the lineage diversified into the 3 extant species, each showing variations on the basic theme. The common vampire bat specializes on large mammals. The white-winged and hairy-legged species prefer birds. These preferences likely reflect differences in foraging behavior, roosting habits, and perhaps sensory specializations for detecting different host types, though detailed comparative studies remain limited. The existence of vampire bats forces reconsideration of what ecological niches are accessible to mammals. Blood feeding is overwhelmingly dominated by arthropods, with only a few vertebrate exceptions, including some finches that peck at seabirds and drink blood, and lampreys that feed on fish blood. Among mammals, vampire bats are unique. They have taken a feeding strategy refined by insects over hundreds of millions of years and re-implemented it using mammalian biology, filling a niche that mosquitoes and ticks normally occupy, but doing so as a warm-blooded, flying mammal with complex social behavior and sophisticated cognition. Whether the specific combination of flight, thermoreception, and rapid amino acid metabolism represents the only viable pathway for a mammal to sustain itself on blood, or whether alternative biochemical or behavioral solutions remain undiscovered, suggests that even well-studied niches may harbor unexplored possibilities.
Through echolocation and evolutionary innovation, bats have shattered the conventional boundaries of mammalian ecology. The spectral bat hunts vertebrate prey like an owl. The fishing bat extracts aquatic prey like a kingfisher. And the vampire bat feeds on blood like a mosquito, each solving problems typically reserved for birds or arthropods through distinctly mammalian solutions. These 3 species reveal a profound truth about evolution. Ecological opportunities remain accessible to any lineage capable of the right innovations, regardless of traditional taxonomic constraints. The sensory sophistication, morphological adaptations, and behavioral flexibility that enable these bats to thrive in unlikely niches demonstrate that nature's solutions are far more diverse than our categorical thinking suggests. As we continue to study these remarkable creatures, we're forced to reconsider what we think we know about the limits of adaptation and the boundaries between ecological roles. Thank you for joining me on this exploration of nature's most versatile aerial predators.